This presentation is a new method for characterizing the complex electrical properties of root segments.
Hello everyone. I'll be talking about the electrical signals of the root segment. In a different meeting, someone pointed at me and said, "This guy is electrifying roots." And some other person said, "Why do you do that?" You'll find out why. The traditional methods for root sampling are partially satisfactory, and the reason is because of the special variability at the root zone, and also because the traditional methods interfere with the natural settings of the root. Because of this, the application of geophysical methods for root investigations has been on the increase. The advantages with geophysical method is high resolution, non-invasiveness is also applicable at the field and also at the laboratory. So many methods has already been used to study the roots at different scales. You see at the root scale, electrical resistance measurement. At the plot scale, you see capacitance measurement, impedance method, impedance spectroscopy. At the plot scale, you see electrical resistivity tomography and electrical impedance tomography, as well as spectral-induced polarization. Between the root scale and the plant scale, they were able to show that conduction and polarization occur at the roots. But then, with ERM, you can only measure the conduction, and at the plant scale, particularly with EIS, they were able to show that polarization takes place at the root. But then the signal you measure, because when you look at the setup, the signal you measure involves the response of the stem, the soil, and the root. So with this method alone, we don't know the response of the root alone. And you look at the plot scale for electrical resistivity tomography. This method has been used to show a correlation between resistivity and root mass density. But what you see here is a bulk response. It contains water, soil water, it contains the root response and also the soil moisture. So it's difficult to separate the response of the root alone. Then also with EIT, recent studies has also used this method to show polarization at the root, but then most of the results are in the bulk root. So the response of the root segments are still missing. So in our experiment, we are going to study the high resolution characterization at the segment scale, 1 to 5 centimeter, and also at a spectroscopic approach. The objective here is to characterize the root segment. Like I said before, the response of the root segment is still missing from the results of the previous studies. So here we are going to show the response of the segment. First of all, we have to design a sample holder that is capable of assessing this electrical response at the segment scale. We are going to also validate the setup and also then study different roots of maize and brachypodium. In this study, our focus is to provide answers to these questions. We want to know if electrical response vary within and across species. We also would like to know what properties of the root influence this electrical response that we measure. Another interesting thing is to find out if it's possible to differentiate the roots from the soil. We look at the measurement setup. We have the sample holder, we use 4 electrode setup, and we have the amplifier unit, the function generator, the data acquisition card, and the computers. The measurement we are taking close to the root collar, and we made sure that the contact between the root and the electrodes are covered in gel. This helps to reduce the influence of contact resistance. And also we made sure that the electrodes were kept out of the current path. Then after the measurement, there is need for data correction. The reason is to minimize errors that result from current leakage. What happens here is that because of the parasitic leakage of the currents, the current injected is not the same as the current that are measured. So there is need to, first of all, in order to correct this, we first take measurement in reverse electrode setting. That means we use the current electrodes as the potentials and use the potential as the current. So when we do this, it enables us to calculate the true excitation current using a MATLAB program. And then with this, we can be able to determine the true sample impedance. This correction helps us to attain an accuracy of 0.1 millirad. Then the calibration, at this stage, we use 3 different ideal resistors. First of all, we calculated the values using the color codes. And then we use the setup to measure again and get the measured values, and we compared them to know if the setup was able to replicate the true values of the resistors. And then we measured on the gel alone and also with a plant. From what we see here, this is the impedance of the gel. This is for 10 kilohertz, 10 kilo ohm resistor, 100 kilo ohm resistor, and then this is 900 kilo ohm resistor. So the measured value matched the calculated values. And here we see the impedance of the root. You can see it's high between the 100 kilo ohm and the 900 kilo ohm. Here we see the polarization, the phase response. We expected that the ideal resistors should not have any phase variation. And here we can see that the 10 kilo ohm resistor, the blue one, there was no phase response. But then the ones with high impedance, there is, at the beginning, we have no phase response. But at high frequency, what we see here is the influence of EM coupling. But you see for the root, this is actually a maze root. What you see here is there is polarization from the low frequency, and it peaks at the high frequency. So we can see that the polarization of the root segment is clearly visible from here. The polarization of the maze root is about 480 millirad. And this is huge when you look at the response of other earth materials, because the range that is usually observed is between 0.2 and 20 millirad. But here we are talking about 480 millirad. Okay, then we look at the current passage in roots. The response of the root when the current passage in the root depends on the frequency. At low frequency, you see that the current will pass through the apoplastic space because the cell membranes inhibit the current flow due to the lipid bilayer. Then the impedance you measure at this stage is mainly the extracellular resistance. Then at high frequency, the cell membrane becomes more conductive, and then the current can pass through the entire system in such a way that the impedance you measure at this level is a combination of both the extracellular and intracellular resistance. And then we look at the polarization in biomaterials. Normally, there are 3 major polarization mechanisms that occur in biomaterials. So first of all, we have the counter ion polarization. Is due to the ionic diffusion at the electrical double layer adjacent to charged surfaces. And you also have the interfacial polarization. What we measure in this case is mainly in the kilohertz range. So that is the then we studied maize and brachypodium plants. We grow the plant in sand because to wash the root, if you wash the root, the water film on the root will interfere with the phase response of the plants. Then we look at the sections of maize and brachypodium in order to be able to explain the variation, what we measure in terms of their electrical response. We see that brachypodium is smaller in size, and while the maize is bigger and has more cells in the cortex, but there's no central metaxylem. Then we see the intraspecies variation. Within species, we see that different maize plants show different response in terms of the resistivity and the phase. The same applies to brachypodium. Then we see the interspecies variability. We see that in terms of the resistivity at day 8, the variation is minimum, but if you look at the phase response, you see that maize showed maximum of 430 millirad, while brachypodium showed 700. And then we see the effect of age. With the age response, we measured over a 4-week period. And for the resistivity, we see an increase and decrease. And also at the phase response, we also see some variation. Maize decreased, and the brachypodium increased and also decreased. In conclusion, we see that intra and interspecies variability occur in the plants, in the roots. Different plants show different response in the variation. We also see that maize and brachypodium, they have unique electrical response that can be used to differentiate them. Then we also see that root properties, the electrical response of the root depend mainly on their age and the anatomy. Then also we can say that when you consider the polarization range of other 8 materials, which is between 0.2 and 20 millirad, then, and you now look at what we measured here, we have 700 millirad for brachypodium and we have 430 millirad for maize. Then it's obvious that the roots can be differentiated from the soil, based mainly on their stronger polarization response. In the next step, we look at the microscopic analysis of the sections to identify the threats that are responsible for this response we measure. And also we are going to look at the hydraulic properties of the roots to study the hydraulic conductivity of the root using our method, and also to see the effect of osmosis. Thank you very much.
Thank you, sir. Are there questions? Yes.